Hello, and thank you for joining us for our MDICX webinar announcing the release of our Fingerstick blueprint for public comment. My name is Carolyn Hiller. I'm the Program Director for our Clinical Diagnostics Initiatives here at MDIC, where it is a privilege to work with the dedicated volunteers that are involved in our work streams. Today, we release the draft version of our Fingerstick blueprint, which was created by the subject matter experts convened in our working group. We need your input, and through April 5th, 2021, we will be accepting public input. The URL will be provided on our closing slide. We'll reserve the last few minutes for questions and answers. Please enter your questions in the Q&A box at the bottom of the screen. Next slide. MDIC is a 501c3 charitable organization that was created for the sole objective of advancing regulatory science of medical devices for public benefit. We do not do policy. Rather, we focus on developing regulatory science tools to help medical device and diagnostic manufacturers navigate. We are member-driven and undertake initiatives that are the priorities of our member organizations. Next slide. It is my pleasure to introduce you to our panelists for today's webinar. Dr. Timothy Stenzel joined the FDA in July of 2018 as Director of the Office of In Vitro Diagnostics and Radiological Health. Dr. Shidhar is Senior Director of Clinical Affairs at Abbott Diabetes Care, and he also serves as the Fingerstick Blueprint Working Group Chair. Dr. Corinne Font is Director of Medical and Scientific Affairs for the Point of Care at Roche Diagnostics Corporation. Dr. Russ Assel is the Acting Lead of the Data Science Team at the Center for Disease Control Prevention. Next slide. And now I'd like to introduce you to Dr. Timothy Stenzel.
Thank you, Carolyn. Pleasure to be here today. Hopefully I can give some background about, why the FDA was very interested in this project and go into some of the challenges that we have faced. So next slide, please. So it became clear, and I'll go into a few of the challenges on this slide, and then some on the next slide, that there was a need for scientifically sound and practical recommendations when evaluating point-of-care devices, including both waived and non-waived devices that utilize these Fingerstick or capillary whole blood samples. And, we had been seeing certain challenges. For example, we had seen performance issues with capillary samples and wanted to see if these issues could be resolved through an effort like this, or at least clarified so that there was a greater likelihood of successful outcomes for devices that come to our office. We'd also seen many submission challenges around study designs and other issues, and we wanted to, sort of harmonize, align on the validations across the various technologies that utilize Fingerstick, both inside the FDA in the different divisions, and with our external stakeholders, including test developers. And you might think, of course, precision studies could be particularly challenging when the sample type is blood from the Fingerstick. So that's kind of a brief background. I want to go into some of the very specific challenges. Next slide, please. So, some of the very advantages of point-of-care testing, that is, it doesn't require a stick for some of these tests, and it is a very small sample volume, creates a challenge for the assay validation. So you've got a small sample type, you know, a single, you know, finger stick yields, you know, sort of a usual amount, and there can be challenges in acquiring that sample collection, you know, from are you getting a sufficient amount, and also subject recruitment. So the subjects that are involved in this study will get their fingers pricked and multiple fingers pricked multiple times, and you know, certainly probably many, if not most of or all of us have had our finger pricked for fingerstick type assay. And it's not nice. It's painful. There isn't necessarily a lot of risk to the patient, but it is uncomfortable. And then, you know, the advantages of point of care devices are, you know, the sample is right there. It doesn't need to be stored. It doesn't need to be shipped, and it can be immediately tested. But then, you know, when you do studies, there are challenges of having stabilized samples. You know, the sample may not be anticoagulated, but if you're going to, you know, do studies, you know, you want to make sure that the sample doesn't clot in collection in your studies. And of course, as with all tests, there's biological variations, and this was impactful on fingerstick studies as well. So I think that's my introduction to this important topic. I do want to close by saying that we really appreciate everybody's input to date who was involved. This is an exciting step forward in making this document public and seeking comment, and we welcome any and all comments. We'll take them seriously, and we welcome the help in advancing this field, and with the goal of really making development of new products that involve finger sticks, which are desirable, more robust pathway that will be easier for all developers, and therefore also make it easier on the FDA review staff to review these applications and authorize them as quickly as possible. So again, thanks to MDIC and everybody who's involved, and especially to you know, the team at CDRH and FDA and in our office who spent so much time on this as well. Thank you. Back over to you, Carolyn, perhaps.
Thank you very much. And Zafar, is my microphone doing better this time?
It is. Thank you.
Okay, great. Let's go to the next slide. I'd like to update everybody of how we came to this process. Now, when the issue was presented to us that there was a desire to have these study designs developed so that there's a more predictable path as you bring your regulatory submissions forward, we convened a group of subject matter experts, not only from the FDA, but also from CDC, our industry, and we had expert advisors that joined us also. Next slide. The team came together and worked through developing the blueprint as it is, and it was a broad group, and we've been working on this for several years. So I want to thank everybody for staying with the project and bringing us to this point. Next slide. Now, to begin with, we wanted to do an assessment and find out where were the pain points. And so we conducted a survey, and we heard from the IVD industry. We also heard from regulatory agencies such as the FDA and Health Canada. We heard from contract clinical research organizations and regulatory consultants. Next slide. And from the results, they said there are two main pain points: precision testing and method comparison. So those are the two tests that the group dove into. Next slide. So in addition to the two performance areas that they're going to talk about in more detail, the blueprint includes definitions and acronyms, regulatory considerations for point of care devices, study designs for candidate devices clearance approval, both for non-waived and CLIA-waived tests, considerations for the use of surrogate samples, considerations for data analysis and method comparison and precision studies, and additional considerations for precision studies with fingerstick samples. Next slide. And I'm going to turn it over to Alva, who will give you more details on precision testing.
Thank you, Garland.
Alva?
Thank you. So I want to thank all the working group members as well as CDRH for the contributions they have made to this document. Can we move to the next slide, please? So the purpose of developing this blueprint, Dr. Stenzel already talked about why we needed this. So this is basically a design of specifically the method comparison and precision for analytical validation of IVD tests. This document is not a comprehensive list of all the things one need to do for a 510(k) approval or a CLIA waiver. This addresses only those two components of the study design, and the requirements for products may vary depending on the design of the product itself, the way the sample is being applied for the test. So there may be some discussions you need to have with the regulatory agency in order for the proper implementation of the designs we have described in this document. As a disclaimer, reading or following this document neither guarantees FDA approval or clearance or payment from any insurance company. So just, this is just a guidance for you to follow as the product is being validated for its analytical performance. Go to the next slide, please. So the intent of the blueprint was to give a direction to the manufacturers on getting a 510(k) approval as well as CLIA waiver. So we try to structure the document such a way that we provide pathways for two different types of approaches. One is a sequential approach where you get a 510(k) clearance first, or de novo or PMA, whatever the classification of the product may be. So you get a clearance of the product first, and then do a CLIA waiver application following the clearance of the product. So you need to do a trained operator study with it for the initial clearance, and with the untrained operator for the CLIA waiver. The second study design is a dual study approach where both the clearance as well as the CLIA waiver are embedded in one study design itself. So you actually have both approaches depending on what the intent of the manufacturer is in terms of getting the clearance as well as a CLIA waiver of the product, whether you want to do in a sequential manner or as all in one kind of an application. Can you go to the next slide, please? So with that in intent, we structured the whole document with, and a lot of effort has been gone into explaining the sequential study design. And there's some reference on how we do a dual study design. All the learnings from the sequential design will be useful for the dual study design as well. So what I'm going to walk you through is the different types of studies we have described in the document, and basically tell you what kind of studies are required. And I will cover the precision study, intent of the precision studies here, and Corinne will come and describe the method comparison and matrix comparison studies later. So for a product which is intended, we'll need to have a fingerstick indication. We need to have a method comparison study done. So where the fingerstick is compared to a comparator method. Fingerstick is done by a trained operator, and the comparator method done is by a laboratory professional. So they are not necessarily the actual operator of the device itself. So in the precision part of the evaluation, there is a reproducibility study done using plasma samples or liquid control, what otherwise we call surrogate samples, and also a component where the reproducibility study is done in the fingerstick sample itself. For that, there are two different approaches we have described. One is what we call design A, where the precision study is embedded in the method comparison study itself, or you do a separate precision study, which will be similar to a method comparison study design, but it's not a component of the method comparison study. And there's also an element of the matrix comparison where you need to show that the fingerstick sample and the surrogate sample performances are similar. So this matrix comparison study can be done by laboratory professionals, either at a clinical site or their own internal site. So we internally termed here as internal site. It can be a clinical site or manufacturer site itself. And there's also evaluation of other components like between lots and between instruments. These don't have to be done in a clinical setting, can be done in this matrix comparison study and the precision study composition, where that study will be done in the internal site. Can you go to the next slide, please? So we also envision that some products may have both fingerstick and venous sample indications. So this study design is actually combining both of them. So you basically do a method comparison study with the fingerstick and venous sample, similar to what we did with the fingerstick. Same way the trained operator does the testing on the candidate device, and there is also a comparator method by the laboratory professional. The precision studies or reproducibility study includes surrogate sample as well as a fingerstick sample and venous sample. Very similar approach. You have a design A and design B, what I described in the previous slide. And also the matrix comparison includes the fingerstick to the venous to the surrogate sample. And you also can do matrix comparison between multiple anticoagulants. So you basically, the method comparison is done with one anticoagulant, and you can do matrix comparison study across multiple anticoagulants. And if there is no difference in the performance between anticoagulants, then you don't have to do a method comparison study again. So that will be a dual indication, fingerstick as well as venous. Again, the disclaimer is we are not talking about heel stick samples and the arterial samples and things like that in this document. They are handled separately. This is specifically focusing on how to get the fingerstick study designs done more consistently. Can we go to the next slide, please? So this is a special situation where if we think that the fingerstick test is not sensitive to sample preparation, or sample collection is not very complicated. So you know, as Dr. Stenzel said, the concern is there's a lot of issue with the sample collection or sample preparation for point of care testing. And if we think the test device is not affected by those methodologies, here is an approach for evaluating a product like that. So the method comparison study is actually done with a venous sample, which we believe is simpler than a capillary sample to be done by the trained operator, very similar to the venous study I described in the previous slide. And the reproducibility study is also done on the venous sample and the surrogate sample. And then the matrix comparison study is done where the fingerstick is shown equivalent to venous sample, because this is a much smaller study design. So we will talk about the matrix comparison study towards the end of the presentation. So you do a matrix comparison between fingerstick and venous, but the method comparison is done with the venous sample. Again, this is a very special situation. You need to consult the regulatory agency for the acceptability of this study design before you actually venture into doing the study like this. So, but the blueprint does provide a provision for a special situation of this type. Can you go to the next slide, please? So in the precision studies, we are basically looking at multiple components of any device. You know, you are all familiar with what all we're looking for. This is basically between run, between day, between lot, between calibration, and any typical product components of variances we're looking for in this study. So if we can go to the next slide. So there is the whole sum of the precision evaluation includes 3 studies, and we call it study 1, study 2, study 3. You know, very simplistic nomenclature. The study 1 is testing the device with the surrogate samples. So these are serum, plasma, or control material. So you can get some of the components like between run, between day, between operator, between site. So these parameters can be evaluated using a surrogate sample. Then in the study 2, you're basically looking at between point of care operator component. So that is using the fingerstick or the venous sample if it's also looking for a venous indication. And the third one is the internal site or the manufacturer site. In addition to the precision between lots and between instruments, differences can be done at the internal site in that study. So basically you have 2 studies done at the point of care test site and 1 study done at the manufacturer test site. That will give you various components of the precision we're looking for. So if you get to the next slide, please. So I talked about design A and design B in the previous slides. So this gives you some flavor for what those studies are. In design A, so in the method comparison study and the precision study are combined, right? So that's what we said. It's one study giving you both. In the study design like this, what we think will happen, or what should happen, is each patient is going to be tested by 2 operators. So you basically have 2 separate finger sticks done on the patient, and the first test is done by the first operator, and the second test is by the second operator and tested on the candidate device. And the results are compared to the laboratory method or comparator method by a laboratory professional. So it's basically the first part is showing you the accuracy study, but you have 2 measurements done by 2 different operators. So you evaluate the precision component between operators from that data. And you also do the rest of the reproducibility study using the standard plasma, serum, or liquid control, or the surrogate sample. So if you go to the next slide, so this is a study where we are not doing the 2 operator test on every study participant. You basically select the concentration ranges for the product. So say your product has 20 milligram to 500 milligram dynamic range. You pick a glucose range, the analyte range between 20 to 70 milligram per deciliter, 70 to 180 milligram per deciliter, or things like that. And then you put 5 patients in each subinterval at each of the point of care site, and those the study participants are tested by 2 operators. So you don't do with every study participant, you do with only 5 participants at each site, and do the study similar to what we described in the previous one, but you basically do 4 measurements rather than actually 2 measurements on those selected study participants. So 2 operators, so each operator does 2 tasks, and that's a study design, which, for some people or some analytes, some devices, you may find the design B is more easier to do than design A. So that selection you can make based on the ease of use of the device as well as the convenience of doing the study. Can we go to the next slide, please? So that's the method validation and the precision combo described for a non-wave test. Now we described the studies for a wave test. So in this case, assumption is you already have done the non-wave test. Now you're doing only the wave test component of that. And you have the study design where there's no comparator. You're basically comparing the untrained operator to the trained operator for the operator comparison. And then you also do comparison of the precision between the trained operator and the untrained operator, similar to the previous study designs. You also have a design A or design B, where you basically do the precision in the operator comparison study plus a precision study as a part of that, or you do it separately. So those are the 2 different options you also have for the comparison method of the precision. So you can go to the next slide, please. So this is a similar design for fingerstick and the venous samples. You do the operator comparison for both the fingerstick sample as well as a venous sample. Then you also do a comparison of the precision with the fingerstick and venous samples by the 2 methods we described, method A and method B. We'll talk about that a little bit later as well. So if you go to the next slide. So This is where it is. So it's basically comparing the two precision study designs for the wave test. In the design A, similar to the wave test, non-wave test. In here, you have each patient is tested by both operators, the trained operator and the untrained operator. So you basically do 4 fingerstick tests per patient. And 3 untrained operators for each site and 2 trained operators for each site is what is described in the document. And you randomize the operator to make sure you don't have an study bias introduced in the study itself. And in the design B, similar to the previous study designs, you have 5 patients per sub-interval of the concentration ranges, and you have 5 fingersticks per patient, 3 by untrained operator and 2 by trained operator, and do this evaluation of the various subcomponents of the precision from that data. So these are the 2 different approaches. Again, as in the previous case, you may want to pick the design based on the complexity of the device and the ease of access to the patients as well. If you can go to the next slide. This is, I think, the only slide we have for the study of a new device where you are seeking both the clear waiver, so basically combining the 510K and the clear waiver in one study. So you basically do a method comparison study. So this is the fingerstick study by untrained operator. If you remember the previous one, you did the 510K with the trained operator and the clear with the untrained operator. In this study, whole of the method comparison study is done by the untrained operator compared to a comparator method by a laboratory professional. The precision evaluation is done by the untrained operator. Similar to the previous one, exactly the same way. You do reproducibility by on the fingerstick sample by 2 of the designs, design A or design B, and you also have a reproducibility study using the surrogate sample. You do matrix comparison as well, similar to the previous study designs we described. So this is basically untrained operator doing all components of the study compared to the previous ones. You had a untrained operator and a trained operator component. Can you go to the next slide, please? So some of the design consideration for the precision studies, the test method you use for doing the precision study has to match the actual use of the product. If you are doing a direct drop to the device, the testing will be done in the direct drop method. If it's going to be pipetted, you pipette the sample for the test device as well. So you basically keep the same method for the precision as well. Obviously, if you're using a surrogate method, you need to adjust the approach slightly differently, because if you're doing a finger stick and doing the direct drop, you cannot do that with the surrogate sample. So you need to modify the methodology. But if you're using for the blood sample, you have to follow the same method as what the intended use of the product is. The test level should cover the measurement range and the medical decision levels. So I've described that a little bit earlier as well. So you need to have full coverage for the dynamic range of the product, and the level should include the medical decision levels when you're assessing the precision. The number of test devices, how many reagent lots, how many test sites you need to have, how many days you need to test the device is all described in the document. And how the data needs to be presented, how the various subcomponents of the variability need to be pulled out of the data and presented in the submission is also provided in the document itself. So we believe this has simplified quite a bit how we are going to look at the data and how you present the data to the regulatory body. So that is my last slide. I'll transfer to Corinne to cover the method comparison and matrix comparison studies for you.
Thank you, Alva. I'd like to thank MDIC for allowing us to be part of this committee, and also the collaboration with the FDA and the other manufacturers who made contributions to this document. So I'm going to go to the next slide, please. And so, Alva already talked about sort of the integration of both the method comparison and the precision studies. I'm going to go into a little bit more detail about the method comparison, so highlighting some of the points he's already emphasized, but then also sharing some other aspects of this part of the document. So let's first start with the non-Waived test, some of the design considerations. This approach really is describing a scenario in which the candidate device is a non-Waived test. We're looking at the combined data set here, and we want the entire measurement range to be spanned in a uniform fashion. So each site really should try to include samples across the measurement range. And so, for example, at 3 minimum test sites, you should really try to spread that testing with about 40 patients at each site. The total of 120 patients minimum should be enrolled for the method comparison for this non-Waived test. Surrogate samples are not recommended for method comparison studies, but we'll get into some details when that may not have an option otherwise. So for these types of studies, at a minimum, we need 3 testing sites with 2 operators per site. So if we can go to the next slide, please. Thank you. This approach describes a scenario in which the candidate device is first cleared or approved as a point-of-care device in the hands of a trained operator, so non-Waived operators. Then a CLIA waiver study is conducted with the objective of comparing the results from the candidate point-of-care device in the hands of untrained operators with fingerstick samples, and the results from the candidate point-of-care device in the hands of trained operators with fingerstick samples. So again, like the non-Waived, the combined data set should have samples that span the full analytical measurement range and approximately uniformly. Each site should try to include the samples across that span of the measuring range. And here, one of the differences is that you need 240 patients minimum to be enrolled for this method comparison for the sequential approach of a Waived test. Surrogate samples, again, are not recommended, and the minimum test sites here is 3, with 1 to 3 untrained operators per site, with a total of 9 untrained operators. So if we can go to the next slide. And here is the approach for the dual approach. This scenario describes a situation when the candidate device is a new device that's not been cleared or approved as a non-Waived point-of-care device. So the studies recommended here provide a single set of comparison and reproducibility studies that the data generated will support both the clearance of the device and the application for CLIA waiver. Again, as with the other 2 examples, the combined data set should span the full analytical measurement range, but here you need 360 total patients to be enrolled for the method comparison for a Waived test. There are provisions provided for situations where only trained operators are allowed to perform the test for some of the sub-intervals. For example, in neonates, you may not be able to put an untrained operator into one of those situations. So it takes into account specific systemic differences between the trained operators and the untrained operators may be necessary to establish that those differences between those 2 groups for specific sub-intervals. Again, all of this really, these are examples provided in the document, but you should confirm with the regulatory agency that will be reviewing your submissions. We can go to the next slide. I'll briefly touch on here in surrogate samples. In general, unmodified patient specimens provide the best assessment for the candidate device performance. However, we recognize that in some cases, it may not be possible to find fresh native samples to cover the extremes of the claimed measuring range. And in some cases, manufacturers can consider modifying the claimed measuring range to an interval that's clinically relevant. Here, as I mentioned before, these unmodified patient specimens are the best assessment of performance. But when they are used, there needs to be evidence that these surrogate samples are used. There needs to be evidence that there's a true representation of the unmodified samples should be provided in your submissions. And different design approaches are provided in the document. And here I want to also emphasize any strategies that you may be thinking of, remember to consult your regulatory agency for acceptability of these different study designs. We'll go to the next slide. If we're going to focus on comparator method. So the comparator method can be any legally marketed device, a predicate device, or any other method that's widely recognized as a reference method, a method that's traceable, or a well-validated method. But in the absence of a reliable laboratory analyzer, another cleared point-of-care device may be used. You need to consider the sample type variations because not all of the comparator methods are validated to use fingerstick samples, and understand the concentration differences between the sample types to adequately address in your these differences in your studies. So we go to the next slide. Here we're going to talk about the data analysis and the presentation. The data range employed in the comparison study should ideally reflect the entire measuring range, as we've mentioned, with an even distribution throughout the measuring range. So that's pretty difficult to do in some of these studies. And one of the problems with comparison studies for fingerstick samples is that potential oversampling can occur at specific subintervals. You know, specifically the normal range sample. So we have an example in the document where clinical study sites such as doctors' offices may include patients with values in the normal range. And oversampling may be unavoidable in the process of trying to get a specific number of balanced patient samples. So we have some ideas in here to minimize oversampling, whether that's within the study design or after the study completion. For example, not performing the comparator tests on those excess samples, performing different types of statistics like weighted regression, or even randomly deleting some of those excess samples for the overfilled bins. And those are all outlined in the document. So if we can go to the next slide, please. And I'm going to finish up here with matrix comparison. So Alva's already mentioned some of the studies that need to be done when you are using, for example, surrogate samples, or if you have specific claims on your device for different sample types. So ideally, performance characteristics of the candidate device with fingerstick specimens should be evaluated with fingerstick patient specimens if at all possible. But matrix comparisons can be done between fingerstick samples and surrogate samples if you've needed to use those for the extreme ends of your analytical measurement range. If you also have claims with venous samples using different anticoagulants, you're going to also need to be doing matrix comparisons. And so at a minimum, it's requested that 40 pairs of results for each sample type covering the full measuring range be done, and repeatability testing 2 replicates are required. And these are all studies that can be performed internal to the manufacturer's site, so on site within the manufacturer's control. So I will conclude there and turn it back over to Carolyn to open it up for questions.
Thank you very much, Corinne, Alva, and Tim. And we do have some questions that have been entered into the Q&A box. Please enter them in and we will work through them. The first question we have is, do you think it will be difficult to get an IRB to approve 5 fingersticks per patient and getting people to actually consent?
This Alva, I can take that question. We believe, there may be some patient population where 5 fingersticks may be difficult to get. It depends on the patient population. We have been successful in some of the IRBs to get up to 10, depending on how you do them, right? If you do with every finger, the 2 sides and things like that. So it depends on the patient population you're targeting for the study itself. So you may have to work with IRB to see what is that they will allow as a valid number of tests doing safely with the patient.
Thank you. Next question, easy one. I'll answer it. Will you email the slide set to meeting attendees? Yes. Thank you for registering. We will send those to you. Next question that we have. Will you please comment on or make recommendations for a test intended for a de novo submission? That is, a test with only an LDT as a comparator method.
I can try. I'm not sure what I think it's a lab test. I'm assuming LDT means. So I think one of the last slides Corinne had described some of that for you. So ideally you need to use a traceable system as a lab analyzer, but in the absence of that, you can use a other POCT method or any other valid method exists. You may want to consult with the regulatory agency or FDA to make sure that is appropriate reference to use for the study itself. So we understand that for every product there is no traceable laboratory analyzer available. So there are provisions made. We have put other point of care device as a possible reference or a comparator method. But if you want to use something other than that, you may want to check with the FDA to make sure that is adequate for you.
Yeah, that's what I would have answered as well, Alva. So thank you.
All right. Okay, next question. You mentioned 5 finger sticks per subject. Do you have any recommendation on how to obtain these finger sticks? Different hand, different fingers, different days? Thoughts?
I can try again. Corinne, you can contradict me if you want. So it has to be on the same day because you're basically testing analyte concentration, which could be potentially varying. So it has to be the same day or basically around the same time. So don't have enough time for the analyte concentration to vary. So it is not the same finger prick. You collect multiple samples to test. So it has to be a unique finger stick for each sample. So you can use different finger, different hand. That should be. There is no reason to believe the left hand and the right hand glucose as an analyte or any other analyte is different based on which hand you take the sample from or which finger you take it from. So we are not making any distinction between the fingers or the hands, but it has to be in a time frame where the analyte concentration doesn't change.
Thank you.
I agree. I agree with that.
Next question. There was mention of using all fingers for studies. Is there recommendations that CLSI GP42 be followed for collection of capillary blood samples?
I would say yes. We are not contradicting any of the existing guidances. Yes.
Okay.
We didn't specifically recommend in the document. I don't recall specifically recommended in CLSI, but yeah, we would have no reason to believe that there's, that you shouldn't follow that for the collection of capillary blood specimens.
Okay. Next comment. Can you comment on the recommended sample size in the method comparison study for a qualitative assay? For example, how many positives would be required?
I don't recall that I don't know, Alva, do you remember if we indicated?
Yeah. We have not considered a qualitative tests as a in the description of the document as much. It is mostly given for a quantitative measurement, because that's where the analytical errors were considered as a problem. So this is primarily to address a quantitative assay. For a qualitative method, you may have other guidances you may want to refer to. So this is specifically for a quantitative assay where the sampling techniques or the number of data points not sufficient to demonstrate the validity of a test is what is addressed in this document.
Okay. Another comment, and this might be a discussion between the different panelists. You usually do not use the thumb or little finger, so you'd need to use only the 6 fingers. Thoughts? Comments?
Yes, that is a standard practice. Thumb is not used or the little finger is not used, and none of our requirements actually goes more than 5 tests, so you still have 1 spare to use. So.
Okay. Next question. Is the proposal for all subjects enrolled in the method comparison study of a fingerstick assay to be subjected to 5 fingersticks?
No, it is only for the ones who are actually in the design B of the precision study. So if they are on the design A of the precision study, there will be 4. If they're in the design B, there will be 5. If they are not in the precision component of the evaluation, then it's only 2. One by 2 operators, so maximum is 2. But it's only the precision component. If you choose to do design B, then you will have a 5 fingerstick requirement.
Thank you. Has any consideration been given for collection of more than 1 capillary tube from a single fingerstick?
No, we talked about it quite a bit, how much sample you can actually get. So we do allow for some pooling of the samples, but not necessarily splitting the sample, one break sample to be split into multiple tasks. We recommend against it, but there is some provision possibly to pool the sample. But yeah, so or you can do a secondary fingerstick. But we felt that collecting too much of capillary sample from one fingerstick may be too excessive.
Okay. Next question. Will this be applied to EUA submissions?
I guess it depends on the timing, is what I would say. You know, we're putting it out for public comment, so it's not a finalized document at this point. And if we're under, you know, emergency use timing, then I don't think there's any reason why it couldn't be if you had a fingerstick test you wanted to put forward.
Thank you. Are there any plans to develop a similar blueprint document, but for home test devices using capillary blood sample collection? I can say we're not going to, at this time, we're not looking at starting a new blueprint. Do one of the panelists want to comment? Would that be considered within the scope of this blueprint, or is it out of it?
Well, the methodologies described here should be adequate for consumer use or the home use products as well. I don't see why we cannot use the guidance provided here for them. So, but there mostly you do the evaluation with the untrained operator anyway predominantly. So I believe the dual approach one will be the one which will be the most appropriate one for a home use testing. So you don't do a sequential. You can still do a sequential, but I think the dual study approach may be more adequate one for home use testing.
Okay. Someone is asking, will the lancet be reused 5 times?
No. No, it will be new lancet, new finger prick.
Okay. Does the blueprint also apply to new, novel, on-the-skin capillary collection devices that are alternatives to a fingerstick?
It did not describe any products of that type in the document anywhere. But if that is the primary capillary sample, and I'm sure it will have the similar concerns in terms of the sample collection approaches and reliability of the test, I think we can extend the learnings from this guidance to that as well.
Okay. How did you determine the sample size for the various studies?
Well, this is in consultation with CDRH. So there is a, for some of the analytes, already a sample size, minimum sample size required documented in various other guidances. So we took them as well as a reference for recommending these kind of sample sizes for them. These are the minimum samples we need where we can statistically show that the data is actually reliable, especially for method comparison. So there is a lot of history in terms of those numbers chosen. We did not make up the number for this document itself. We took the other references existing to recommend these sample types here, sample sizes here.
Okay. Which study design approach should a fingerstick collection device, not a POCT, follow, waived or non-waived?
I'm not sure I understand that question.
Yeah, so because this guidance or waive or non-waive applies to mostly analytical tests, not for collecting the sample, as I understand.
Yeah, not the collection devices, right? So yeah, it's for, like, for example, a point of care glucose meter, not fingerstick collection devices.
Okay. Have specific analytical and modeling methods that should be used for conducting proper statistical analysis of the resultant data been addressed in the blueprint?
So yeah, go ahead, Alba.
So you go ahead.
I was just going to say, we worked, hand in hand with the FDA and their biostatistician for this document. And so, all the linear regression modeling statistics that are described in the document are sort of accepted and well validated with the agency. So, of course, any study design that you want to do that may be outside of this or different should be, you should consult the FDA for acceptability with your study design. But there are specifics listed within the blueprint.
Yes.
Thank you. I think we have got the questions answered in the box. Your last chance to put up some questions. Otherwise, you can see on your screen where you can download the document. We ask for you to, in your response, to give the line number. What is your issue with how it is phrased? And most importantly, we look for your suggestion for how we can resolve the issue. We have one more question that I'm going to take. From what was discussed here, can we say that in precision studies, surrogate samples should only be used for reproducibility only, and fingerstick sample for operator, and all other studies?
There's not all of it. In some studies, there is an operator-to-operator comparison as well. Yes, you can go through each study design and the different approaches are described there. But for surrogate sample is allowed only for precision studies.
Well, unless there's in the method comparison where you don't have a choice, right?
That's correct, yeah.
Yeah, so if you don't have a choice, they'll be allowed. But again, those alternative designs really should be put forth with the agency to have those discussions.
Yes.
Okay. Again, I want to thank our working group volunteers and today's panelists and all of you for participating in our teleconference today. Please, we look forward to your input so that we can make this a valuable regulatory science tool for you. We will be sending out the slides. Thank you very much for joining us today. Please be well. Goodbye.
Thank you.
Thanks.